There is something your body has been doing since the moment you were conceived, something so quiet, so constant, so deeply woven into the fabric of every cell you carry, that you have lived your entire life inside its protection without ever being given a name for it. You have felt it without knowing what it was. You have benefited from it without anyone telling you it existed. And if you are a woman, particularly if you are a woman over 40, over 50, over 60, you have been living proof of it for decades. Think about this for a moment. Think about the women you have known in your life. Think about your mother, your grandmother, your great-grandmother, if you were lucky enough to know her. Think about the pattern that was so consistent, so ordinary, so woven into the background of your family's story that you probably never stopped to question it. The women lasted longer. The women were still standing when the men were gone. Your grandmother buried your grandfather and then lived another 10, 15, 20 years. Your mother outlived your father. Your great-aunt was the last one alive at every family gathering, sitting in her chair, sharp-eyed, remembering everything, the living archive of a family whose men had already been lowered into the ground. You probably assumed this was coincidence, or lifestyle, or the fact that the women in your family took better care of themselves, ate better, worried less, went to the doctor more, drank less, smoked less, took fewer stupid risks, and some of those things may be partly true in certain cases in certain generations. But they cannot explain what is actually happening. They cannot explain why this pattern holds in every single country on Earth. Not most countries, every country, in every era that has ever been recorded, under every condition humanity has ever endured. Wealth and poverty, peace and war, feast and famine, modern medicine and no medicine at all. They cannot explain why the pattern holds not just in humans, but in baboons, gorillas, elephants, whales, and the vast majority of mammalian species that have ever been studied. They cannot explain why the pattern shows up before birth, why male fetuses are more likely to be miscarried, why male newborns are more likely to die in their first hour, their first day, their first week, their first month, their first year, before any baby has made a single decision about diet, exercise, strategy, stress management, or risk tolerance. The pattern is not behavioral. It is not cultural. It is not a product of modern healthcare or lifestyle choices, or the fact that women are more careful. The pattern is built into the body. Your body, the body you wake up in every morning, the body you have carried through pregnancies and illnesses and losses and recoveries and decades of labour that no one measured or compensated. That body was engineered, at the deepest level biology can reach, to outlast the male body, not by a little, not as a minor statistical fluctuation, by years, by decades, at the extreme end. 85% of every human being who reaches 100 years old is a woman. Over 95% of every human being who reaches 110 is a woman. The oldest verified person in the history of the species, Jeanne Calment of France, who lived to 122 years and 164 days, was a woman. The second oldest was a woman. The third oldest was a woman. Of the 10 oldest humans ever reliably documented, all 10 were women. This is not coincidence. This is architecture. And tonight, we're going to walk through that architecture. Not the shallow version you have heard before. Not the version that says women go to the doctor more and men take more risks. The deep version. The version written in your chromosomes, your hormones, your mitochondria, your telomeres, your immune cells, your fat tissue, your stress hormones, your cellular repair mechanisms. The version that took 300,000 years of relentless natural selection to build. The version that no one ever told you about, because for most of recorded history the people studying biology were men, and they were studying the male body as the default, and the female body was the variation, the deviation, the softer version, the weaker version, even as that so-called weaker version was quietly, systematically, generation after generation, outliving the so-called stronger one. If you have ever felt that your body was underestimated, that its strength was invisible, its endurance unacknowledged, its survival unvalued, then what follows is the deepest vindication biology can offer. Your body was never fragile. It was never the lesser model. It was never the compromise. It was the version that evolution spent 300,000 years perfecting. Because your survival, more than any other variable in the entire human story, was the one thing the future of the species could not afford to lose. This is the 300,000-year answer, and it has been hiding inside your cells your entire life. She did not have a name that history recorded. She lived somewhere in East Africa, roughly 300,000 years ago, in a world that would be unrecognizable to you in almost every way except one. The world was trying to kill her, and she was refusing to die. She woke before dawn. The air was cool. The child sleeping against her chest shifted but did not wake. Her body ached, the specific ache of a woman who has carried weight all day across uneven ground, who has crouched for hours digging roots from hard soil, who has nursed a child through the night while sleeping on the earth. The predators were still a possibility in the grey half-light. The water source was a walk away. The food she'd gathered yesterday was nearly gone. She did not know the word chromosome. She did not know she carried 2 X chromosomes, and that this gave her a genetic backup system for over 1,000 critical genes. A system that the man sleeping a few meters away did not have. She did not know the word estrogen, or that this molecule was, at that very moment, maintaining the elasticity of her blood vessels, activating telomerase in her cells to protect the ends of her chromosomes from degrading, reducing the inflammatory damage accumulating in her tissues, and shielding her heart from the atherosclerotic buildup that was already beginning, silently and irreversibly, in the man's arteries. She did not know the word mitochondria, or that the tiny energy-producing organelles inside every one of her cells, the engines powering every heartbeat, every thought, every step she was about to take toward water, had been optimized by millions of years of evolution for her body specifically, tested and refined exclusively through the female line, and that the man's mitochondria, inherited from his mother, were running on hardware that had never been quality tested in a male body. She did not know the word telomere, or that the protective caps on the ends of her chromosomes were measurably longer than his, set at a higher starting point from the moment of her conception, and that this difference would translate across her lifetime into cells that age more slowly, organs that maintained function longer, and a body that was biologically younger than his at every shared birthday from the cradle to the grave. She did not know any of this. She knew how to find water. She knew which roots were edible and which would make her sick. She knew the sound that meant a predator was close and the sound that meant it had moved on. She knew how to keep a child alive. She knew how to keep herself alive. And she kept doing it, day after day, season after season, year after year, longer than he did. Because her body was built to. And because she kept living, so did we. This is the question we're answering tonight. Not the polite, simplified version. Not the version that fits on a magazine cover or in a 2-minute news segment. The deep version, the complete version. The version that begins in the genome and moves outward through every system in the female body, layer by layer, mechanism by mechanism, until you understand, not as an abstraction, but as a physical reality you can feel in your own cells. Why the female body was engineered for endurance at a level that the male body was not, is not, and likely never will be. The question of why women outlive men is not new. Demographers and physicians have known the pattern for well over a century. In every country that keeps reliable records, and many that do not, but where researchers have pieced together historical data from parish records, cemetery inscriptions, and colonial administrative archives, women live longer than men. In the United States today, the gap is approximately 5 to 6 years. In Japan, it is roughly 7. In Russia, it stretches past 10. In every country in the European Union, in every nation in sub-Saharan Africa, in every country in South America, in every island nation in the Pacific, in every population that has ever been systematically studied, women outlive men. Our World in Data, one of the most comprehensive demographic research platforms in existence, states it with the simplicity of an obvious truth. Women live longer than men in every country in the world. Not most, not the majority, every single one. For most of the century that this pattern has been formally documented, the explanation stayed shallow. Men take more risks, men die in wars, men work in more dangerous jobs, men smoke more, drink more, fight more, drive faster, visit the doctor less, suppress their emotions, ignore their symptoms, refuse to ask for help. These behavioural explanations are not entirely wrong. In modern industrialised societies, behavioural differences between men and women do contribute to the gap. Men are more likely to die from accidents, homicides, drug overdoses, and alcohol-related causes. Men are less likely to seek preventive care. Men are socialised in many cultures to treat vulnerability as weakness, which delays treatment of serious conditions. But behavioural explanations have a fatal fundamental, inescapable flaw. They cannot explain the biology. They cannot explain why the female survival advantage appears before birth. In the womb, the story has already begun. Male fetuses are more likely to be spontaneously aborted. More male pregnancies end in miscarriage. Male fetuses are more susceptible to complications during gestation. At birth, say more boys are born than girls. The sex ratio at birth is approximately 105 boys for every 100 girls, and biologists believe this slight excess of male births exists precisely because nature is compensating in advance for the higher male mortality that will follow. More boys are produced because more boys will die, and die they do. More male newborns die in their first 24 hours of life. More die in their first week. More die in their first month. More die in their first year. This is true in the wealthiest countries with the most advanced neonatal intensive care units in the world, and it is true in the poorest nations with almost no medical infrastructure at all. In neonatal intensive care units around the world, premature female infants survive at higher rates than premature male infants of the same gestational age and birth weight. A 2011 study published in the Journal of Perinatology found that after controlling for body weight, gestational age and size, female preterm infants were significantly more likely to survive, 95.4% versus 93.6% for males. A 2015 Loyola University study provided further evidence that premature girls not only survive better, but reach developmental milestones faster. They orally fed independently a full day earlier than premature boys. These are newborns. They have no lifestyle. They have no behavioral patterns. They have made no choices about diet, exercise, risk tolerance, or healthcare utilization. They are simply female. And that fact alone, written into every cell at the moment of conception, is enough to tilt the odds in their favour. In 2018, a research team led by Virginia Zaruli of the University of Southern Denmark and James Vorpal of Duke University published a landmark study in the Proceedings of the National Academy of Sciences that should have permanently ended the behavioural explanation as a sufficient answer. The researchers examined 7 populations that had endured the most catastrophic mortality conditions in recorded human history. Conditions so extreme that average life expectancy had collapsed to 20 years or less, and in some cases to less than 3 years. The populations were the freed American slaves who migrated to Liberia between 1820 and 1843, more than 40% of whom died in their first year from tropical diseases they had no resistance to, and whose babies rarely survived past their second birthday. The plantation slaves of Trinidad between 1813 and 1816, whose lives were controlled entirely by the economic decisions of slave owners. The victims of the Ukrainian famine of 1933, when Stalin's forced collectivization deliberately starved millions of Ukrainians to death, and life expectancy collapsed to single digits. The Swedish famine of 1772 to 1773, the Icelandic measles epidemics of 1846 and 1882, and the Irish potato famine of 1845 to 1849, when a blight destroyed the staple crop and more than 1 million people starved to death. In every one of these populations, starving, enslaved, disease-ravaged, dying in numbers that defy comprehension, women outlived men. In Ukraine in 1933, male life expectancy fell to 7.3 years. Female life expectancy was 10.85 years, nearly 50% higher. In Ireland during the famine, men averaged 18.7 years, women 22.4, nearly 4 years longer. In Liberia, where mortality was so extreme that men's life expectancy was 1.68 years, less than 2 years, women's was 2.23 years. Even when life itself was measured in months, women had more of them, and the most devastating finding, the one that should permanently silence anyone who claims the gap is behavioural, was this: in most of these populations, the single largest contributor to the female survival advantage was not adult hardiness, it was infant survival. Newborn girls survived these catastrophes at higher rates than newborn boys. In Liberia, 77% of the total life expectancy gap came from the 0 to 1 age group. In Trinidad, it was as high as 88%. In Ireland, 67%. Babies, hours old, days old, weeks old, making no choices, having no behaviours, simply female. and more likely to survive the worst conditions humanity has ever inflicted upon itself. Virginia Zarulli and her colleagues were careful in their language, as scientists must be. But the conclusion was unmistakable. The female survival advantage cannot be fully explained by behavioural or social differences. It has fundamental biological roots. That is where this documentary begins. Not in behaviour, not in culture, not in who goes to the doctor more or who takes fewer risks. In biology, in the body, in the architecture of survival that the female body carries in every chromosome, every hormone, every mitochondrion, every immune cell, every telomere, every fat cell, every neurochemical pathway. An architecture assembled over 300,000 years of Homo sapiens evolution and over hundreds of millions of years of mammalian evolution before that. An architecture that no one ever told you about. An architecture that, by the time you finish listening tonight, you will understand at a depth that changes how you think about your body, your mother's body, your grandmother's body, your daughter's body, and the bodies of every woman who has ever lived. The first layer of that architecture is the most fundamental thing about you. It was determined at the instant of your conception. It is present in every cell in your body, in your brain, your heart, your liver, your skin, your blood, your bones. It is the reason that the very first line of the survival story is already written before you take your first breath. It is your chromosomes, and the story they tell is extraordinary. Chapter 1. The Genetic Architecture of Endurance. Every human being is built from a set of instructions written in DNA, distributed across 23 pairs of chromosomes. 22 of those pairs are the same in men and women. They are called autosomes, and they contain the blueprints for the vast majority of your body's structures and functions. Your organs, your bones, your muscles, your metabolic systems, the colour of your eyes, the curl of your hair. But the 23rd pair is different. It is the pair that determined, at the instant of conception, whether you would develop as male or female. And the difference between the two possible configurations of that pair, XX for female, XY for male, is not a minor footnote in human biology. It is one of the most consequential genetic facts in the entire story of human survival. To understand why, you need to understand what the X chromosome actually is. The X chromosome is not a small chromosome. It is not a minor player in the genetic orchestra. It is one of the largest chromosomes in the human genome, carrying approximately 1,100 genes that code for proteins involved in immune function, brain development, blood clotting, muscle repair, organ maintenance, neurological signaling, cellular metabolism, and dozens of other systems that are essential for staying alive. The X chromosome is, in a very real sense, one of the most important survival chromosomes in the human genome. Now compare it to the Y chromosome. The Y chromosome, the chromosome that triggers male development, is tiny. It carries roughly 50 genes, 50 compared to the X chromosome's 1,100. It is one of the smallest chromosomes in the human genome, and over the course of hundreds of millions of years of evolution, it has been steadily shrinking. It cannot recombine with a second copy of itself, because there is no second Y in male cells, and this means it cannot repair itself through the same mechanisms that other chromosomes use to correct accumulated damage. It accumulates mutations. It loses genes. Some evolutionary biologists have described the Y chromosome as being in a slow, irreversible process of genetic decay. This is not speculation. The Y chromosome has already lost over 90% of the genes it once shared with the X chromosome. In certain rodent species, the Y chromosome has disappeared entirely, and those species have evolved alternative mechanisms for sex determination. Whether the human Y chromosome will eventually follow the same path is debated. Some researchers estimate it could disappear within a few million years, others argue it has stabilised, but the trajectory is clear. The chromosome that defines maleness is the most fragile, the most degraded, and the most genetically impoverished chromosome in the human genome. The chromosome that defines femaleness is one of the most robust, the most gene-rich, and the most functionally critical. And because women carry 2 copies of it, they carry a survival advantage that men cannot access. This is what geneticists call the unguarded X hypothesis, though calling it a hypothesis almost understates its evidentiary support because it has now been confirmed across over 1,000 species. The logic is straightforward. Every chromosome accumulates mutations over time. Some of those mutations are harmful. They produce defective proteins, or they fail to produce essential proteins at all. When a harmful mutation occurs on one of a woman's 2X chromosomes, the second X chromosome can compensate. The functional copy of the gene on one X masks the broken copy on the other. The woman's cells continue to produce the protein they need. She may never know the mutation exists. When a harmful mutation occurs on a man's single X chromosome, there is no backup, no second copy, no masking. The mutation expresses fully. The protein is defective or absent. The man suffers the consequence. This is why the vast majority of X-linked genetic disorders overwhelmingly affect men. Haemophilia, the inability to clot blood properly, which can cause life-threatening bleeding from minor injuries, is caused by mutations on the X chromosome. It affects almost exclusively men. Duchenne muscular dystrophy, a devastating condition that causes progressive muscle wasting, typically killing patients in their 20s or 30s, is X-linked and affects almost exclusively men. Red-green colour blindness, which affects roughly 8% of men and less than 0.5% of women. Certain severe immunodeficiency syndromes. Certain forms of intellectual disability. The list goes on. Hundreds of X-linked conditions that are overwhelmingly or exclusively male diseases, not because men are genetically inferior, but because they are genetically exposed. They have one copy of a critical chromosome where women have two. In 2025, an international research team led by scientists at the Max Planck Institute for Evolutionary Anthropology published the most comprehensive analysis of sex differences in lifespan across mammals and birds ever conducted. Using records from over 1,176 species in zoos worldwide, where behavioral and environmental variables are controlled, they found that in 72% of mammal species, females lived longer than males. The average female advantage was 12%. In birds, where the chromosome system is reversed, males carry two matching Z chromosomes while females carry one Z and one W. The pattern flipped. In 68% of bird species, males lived longer. The chromosomes predict the direction of the longevity gap across the entire animal kingdom. The sex with two matching sex chromosomes, the sex with the genetic backup system, lives longer. In mammals, that is the female. In birds, that is the male. The study, published in Science Advances, confirmed what had been suspected for decades. The survival advantage is chromosomal. It is structural. It is embedded in the most basic genetic fact about how males and females are built. Some species show the opposite of the expected pattern, lead author Johanna Sterk acknowledged. For example, in many birds of prey, females are both larger and longer-lived than males, so sex chromosomes can only be part of the story. She is right. Sex chromosomes are not the whole story, but they are the first chapter, the foundation on which every other layer of the female survival advantage is built. But the XX advantage goes deeper than simple backup. There is a second mechanism, more subtle, more recently understood, and in some ways even more remarkable, that gives women a form of biological resilience that men cannot possess. It is called X chromosome inactivation, and it creates something extraordinary, a female body that is a genetic mosaic. Early in embryonic development, one of the two X chromosomes in each female cell is randomly silenced. This process, discovered by geneticist Mary Lyon in 1961, is called lyonization. In each cell, either the X inherited from the mother or the X inherited from the father is inactivated. This silencing is random and permanent for that cell and all of its descendants. The result is that, across a woman's body, roughly half of her cells are using the maternal X and the other half are using the paternal X. She is, in a very literal sense, two different genetic individuals woven into one body. This mosaic has profound implications for survival. If a harmful mutation exists on one X chromosome, only the cells where that particular X is active will be affected. The rest of the body, where the other, healthy X is active, continues to function normally. This means that diseases, dysfunctions, and vulnerabilities that would devastate a man's entire body, because every one of his cells is running the same single X, may affect only half of a woman's cells, with the other half functioning as normal. The mosaicism gives women internal genetic diversity. Different cells in the same organ may be running slightly different genetic programs. Different immune cells may be producing slightly different versions of key proteins. Different metabolic pathways may be operating at slightly different efficiencies. This diversity is not a flaw. It is a feature. It gives the female body more options, more flexibility, more ways to respond to threats. If one genetic program fails, the other may succeed. If one version of a protein is less effective against a particular pathogen, the other version may be more effective. A man's body has no such diversity on the X chromosome. Every cell runs the same single program. There is no mosaic, no variation, no backup strategy. Researchers at Frontiers in Young Minds published an accessible explanation of this principle in 2019 titled X Marks the Spot: How X Chromosome Inactivation Gives Females a Genetic Advantage. The article stated, cellular mosaicism gives females more diversity than males, meaning that they have more options during development and more ways to prevent disease. More options, more ways. This is the language of engineering redundancy, the same principle that makes aircraft safe, that puts two engines on a plane when one would suffice for flight, that duplicates critical systems so that the failure of one does not mean the failure of all. 2. Your body is built with the redundancy principle of an aircraft. 2 X chromosomes, 2 programs, 2 options. A mosaic of genetic strategies distributed across every organ, every tissue, every cell line. And the male body has 1 X chromosome, 1 program, 1 option, no mosaic, no backup. This is the genetic architecture of endurance, and it is only the first layer. Beneath it, working in concert with it, amplifying its effects across every decade of life, there is a second system, a chemical system, a hormonal system, that takes the chromosomal advantage and extends it into every blood vessel, every brain cell, every organ in the body. And that system is dominated by a molecule that the modern world has spent decades both celebrating and fearing, without ever understanding what it was truly built to do. Chapter 2. The estrogen shield. There is a molecule flowing through your blood right now, if you're a premenopausal woman, that is doing more to keep you alive than almost any other single substance in your body. It is repairing the walls of your blood vessels. It is reducing the buildup of dangerous plaque in your arteries. It is lowering your blood pressure. It is raising the protective form of cholesterol in your blood while lowering the harmful form. It is activating an enzyme in your cells that maintains the protective caps on the ends of your chromosomes, slowing the fundamental clock of cellular aging. It is modulating your immune system to fight infection more effectively. It is protecting your neurons from the inflammatory damage that drives cognitive decline. It is maintaining the density of your bones. It is supporting the elasticity of your skin. It is doing all of this simultaneously every hour of every day, and it has been doing it since your body first began producing it in significant quantities during puberty. That molecule is estrogen, and what you are probably taught about it, that it is a reproductive hormone, that its primary purpose is to regulate the menstrual cycle and support pregnancy, is not wrong, but it is catastrophically incomplete. It is like describing the sun as a source of light. Yes, the sun produces light, but the sun also produces the heat that makes life on Earth possible, the gravitational field that keeps the planets in orbit, the solar wind that shapes the entire heliosphere. Calling it a source of light is technically accurate and functionally misleading. Estrogen is a survival hormone, so reproduction is one of its functions. Keeping you alive is the larger one. The cardiovascular protection alone is so dramatic that it reshaped the entire field of cardiology once researchers finally noticed it. For most of the 20th century, heart disease was considered a man's disease. The image of the heart attack victim, clutching his chest, collapsing at his desk, rushed to the emergency room, was overwhelmingly male, and for good reason. Men in their 40s and 50s suffer heart attacks at rates that dwarf those of women the same age. The gap is not small. A 40-year-old man's risk of a coronary event is multiple times higher than a 40-year-old woman's. A 50-year-old man's risk is still dramatically higher. The American Heart Association, the British Heart Foundation, and every major cardiology organization in the world recognizes that premenopausal women have a level of cardiovascular protection that premenopausal men simply do not. That protection is estrogen. Estrogen relaxes blood vessel walls through the production of nitric oxide, a vasodilator that improves blood flow and reduces the mechanical stress on arterial walls. It raises HDL cholesterol, the form that acts as a scavenger, removing dangerous deposits from artery walls and transporting them to the liver for disposal. It lowers LDL cholesterol, the form that infiltrates artery walls and forms the fatty, inflammatory plaques that cause heart attacks and strokes. It reduces the expression of adhesion molecules on the inner lining of blood vessels, making it harder for white blood cells and platelets to stick to vessel walls and initiate the inflammatory cascade of atherosclerosis. It modulates the clotting cascade, maintaining the delicate balance between clot formation and clot prevention that prevents both hemorrhage and thrombosis. The Mayo Clinic's research on estrogen and heart disease is explicit. Dr. Delisa Fairweather states that estrogen helps protect against heart attack by preventing the inflammation that contributes to the buildup of plaque in the arteries. This is not a secondary effect. This is not a minor benefit. This is the primary reason that premenopausal women have a fraction of men's heart attack risk. The molecule that the modern world associates primarily with fertility and menstruation is, in reality, one of the most potent cardiovascular protectants in all of biology. And then menopause arrives, and the shield drops, and the proof becomes devastating. After menopause, when estrogen production declines by roughly 90%, women's cardiovascular risk rises sharply. Within 10 to 15 years after menopause, women's rates of heart attack and stroke approach, and in some populations equal, men's rates. The same woman, living the same life, eating the same food, exercising the same amount, carrying the same stress, sees her heart disease risk surge. The variable that changed was not behavioural, it was hormonal. Estrogen fell, and the heart lost its shield. This is one of the most powerful pieces of evidence in all of sex-based medicine. It is a natural experiment replicated in every woman who passes through menopause. You are demonstrating that estrogen, not lifestyle, not attitude, not healthcare access, is the primary engine of the premenopausal female cardiovascular advantage. When estrogen is present, the heart is protected. When estrogen withdraws, the heart is vulnerable. The molecule drives the outcome, but the estrogen shield does not stop at the heart. Its reach extends into every cell in the body, through a mechanism that connects hormones to the most fundamental clock of biological aging. Telomeres, the protective caps on the ends of chromosomes, shorten with every cell division. When they become critically short, cells stop dividing and enter senescence, a state of permanent dysfunction in which they release inflammatory signals that damage surrounding tissue. The accumulation of senescent cells drives aging in every organ. It is one of the central mechanisms of why bodies deteriorate over time. Estrogen activates telomerase, the enzyme that can add new repetitive sequences to telomere ends, counteracting the shortening process. This was confirmed in a 2022 study published in Aging Cell, which demonstrated that female telomeres are measurably longer than male telomeres, between 100 and 300 base pairs longer, and that this difference is already present at birth. The researchers traced the birth difference to higher levels of a protein called discarin in female embryos. Discarin is essential for telomerase function. The gene that produces discarin, DKC1, is located on the X chromosome. In female embryos, both X chromosomes are briefly active before X inactivation occurs, producing more discarin during the critical developmental window when telomere length is being set for all the stem cells that will populate the body for its entire lifespan. Female embryos produce more telomere-building protein. They start life with longer telomeres, and then estrogen maintains that advantage for roughly 35 to 40 years by continuously activating telomerase throughout the reproductive years. The researchers calculated that the telomere length difference at birth corresponds to a 5 to 8 year difference in average life expectancy. 5 to 8 years. That is remarkably close to the actual observed gap in most developed nations. The cellular clocks are set differently from the moment of conception, and the hormonal environment maintains and extends that difference across the entire reproductive lifespan. Men receive the opposite hormonal treatment. Testosterone, the molecule that defines much of male physiology, is, from a longevity perspective, spectacularly expensive. Testosterone builds larger muscles, denser bones, thicker skin, a deeper voice, and the capacity for explosive physical output. In ancestral environments, these traits were essential for survival, for fighting, hunting, protecting territory, competing for mates. But they come at a biological cost that is paid in years of life. Testosterone suppresses immune function. It promotes chronic low-grade inflammation. It accelerates the development of arterial plaque. It drives the higher metabolic rate that comes with maintaining larger muscle mass, which in turn generates more oxidative stress. More free radicals, more cellular damage, more accumulated wear on every organ. Testosterone is associated with higher rates of cardiac hypertrophy, the thickening of the heart muscle that can lead to heart failure. It is associated with more aggressive risk-taking behavior, which increases injury and death. and its metabolic demands mean that male cells divide more frequently to maintain larger tissues, shortening telomeres faster. The evidence from Korean eunuchs is among the most striking in all of longevity research. In 2012, researchers led by biologist Kyungjin Min at Inha University in South Korea analysed historical records from the Chosun dynasty, records that meticulously documented the lives of eunuchs, men who had been castrated before puberty and therefore produced virtually no testosterone for the rest of their lives. The eunuchs lived an average of 70 years. Non-castrated men of the same socioeconomic class lived an average of 50 to 56 years. The eunuchs outlived their intact counterparts by 14 to 19 years. 3 of the eunuchs in the records lived past 100 years, at a time when reaching 100 was extraordinarily rare for anyone. 14 to 19 years. That is not a small difference. That is the difference between dying at 55 and living to see your great-grandchildren, and the only variable that changed was testosterone. Remove the male sex hormone, and male lifespan approaches, and in some cases exceeds, female lifespan. Our study supports the idea that male sex hormones decrease the lifespan of men, the researchers wrote. This is diplomatic scientific understatement. What the data actually shows is that testosterone is one of the single most life-shortening substances in the male body, a molecule that trades years of life for the physical traits needed to compete and reproduce. 20th century records of institutionalized men who had been castrated showed a similar pattern. Extended lifespan compared to intact men. The evidence converges from multiple populations, multiple centuries, multiple methodologies. Estrogen extends. Testosterone costs. The hormonal environments of the female and male bodies are not mirror images. They are opposite survival strategies. Estrogen is an investment in durability, a long-term maintenance contract for the body's infrastructure. Testosterone is an investment in performance, a short-term boost to the body's competitive capacity. Both strategies served evolutionary purposes, but they have different price tags, and the price of testosterone is paid in years that men do not live. For most of human history, the average woman lived with estrogen's protection from her early teens until her late 40s or early 50s. That is roughly 35 to 40 years of cardiovascular shielding, telomere maintenance, anti-inflammatory defense, neuroprotection, bone density maintenance, and cellular repair enhancement. 35 to 40 years of quiet, continuous maintenance work happening inside every blood vessel, every neuron, every organ, every cell. Work that the male body, running on testosterone's expensive fuel, is not receiving. By the time menopause arrives and the shield begins to lower, the accumulated advantage is enormous. Decades of fewer senescent cells. Decades of less arterial plaque. Decades of longer telomeres. Decades of less oxidative damage. The male body, by the same age, has been running without that maintenance for its entire life. The damage has compounded. The organs have aged faster. The gap cannot be closed. If you are a woman past menopause, you may feel that estrogen's departure has left you more vulnerable. And it has, relative to where you were. But relative to a man of the same age, you are still carrying the accumulated capital of decades of cellular maintenance that his body never received. The shield may have lowered, but the fortress it built is still standing, and underneath the estrogen shield there is another system, older, fiercer, and more immediately consequential for daily survival, that gives the female body yet another layer of protection. A system that fights harder, responds faster, and remembers longer than its male counterpart. A system that has kept women alive through every plague, every epidemic, every pandemic in the 300,000-year history of our species. Chapter 3. The Immune Fortress. In the year 1918, a virus swept across the world and killed somewhere between 50 million and 100 million people, more than the First World War that was ending at the same time. The Spanish influenza pandemic was the deadliest disease event in modern human history. It struck with terrifying speed. Healthy young adults could develop symptoms in the morning and be dead by evening. Hospitals overflowed. Mass graves were dug in city parks. Entire communities were wiped out, and men died at higher rates than women. A study analysing Kentucky death certificates from 1911 to 1919, published in the Journal of Infectious Diseases, found that during the fall 1918 pandemic wave, the most lethal wave, excess mortality rates were 20 to 80% higher among young adult males aged 15 to 50 compared to females of the same age. Nearly 175,000 more men died than women in the United States in 1918. Epidemics, the data showed, tend to kill more men than women. The 1918 flu was not an exception. It was the rule. A century later, in 2020, the pattern repeated. When SARS-CoV-2, the virus that causes COVID-19, spread across the world, men died at higher rates than women in 33 of 35 countries that reported sex disaggregated data. The male-to-female death ratio was consistently above 1, meaning more men died, across almost every age group, every country, and every phase of the pandemic. Studies published in journals from The Lancet to Nature confirmed what the 1918 data had already shown. When a novel pathogen attacks the human species, the female body fights it off more effectively than the male body. This is not limited to respiratory viruses. Women have lower mortality rates from tuberculosis, hepatitis B, HIV, Ebola, malaria, and virtually every major infectious disease that has been studied with sex disaggregated data. Women clear infections faster. They mount stronger antibody responses. They build more durable immunological memory. They survive. The reason is the immune fortress. The female immune system is not just slightly better than the male immune system. It is built differently, assembled differently, regulated differently, and powered differently at every level. From the chromosomes that code for it to the hormones that modulate it to the individual immune cells that carry out its work. It begins, once again, at the X chromosome. The X chromosome carries a disproportionate number of immune-related genes. Genes for toll-like receptors that detect pathogens, genes for cytokines that coordinate immune responses, genes for antibody production, genes for T-cell function, genes for the complement system that tags invaders for destruction. Women, with 2 X chromosomes, carry 2 copies of these genes. Men with 1 X carry 1. But the advantage is amplified by the mosaicism we discussed in the previous chapter. Because of X chromosome inactivation, different immune cells in a woman's body may be expressing different versions of these immune genes. Some of her T cells are running the maternal X program, others are running the paternal X program. This gives her immune system a form of internal diversity, a broader repertoire of immune responses that the male immune system, locked into a single X chromosome, cannot match. Researchers at the La Jolla Institute for Immunology, one of the world's leading centers for the study of immune function, have described the female immune advantage as consistent and measurable across all age groups. Women carry more CD4 plus T cells, the master coordinators of the adaptive immune response, the cells that orchestrate the attack against specific pathogens. They carry more B cells, which produce antibodies. They carry more plasma cells, which are the long-lived factories of antibody production that provide lasting immunity after infection or vaccination. They carry more regulatory T cells, which modulate immune responses to prevent overreaction. They carry more of the specialized immune architecture that identifies, targets, remembers, and destroys threats. The functional differences are not subtle. In mouse models of sepsis, the life-threatening systemic infection that kills hundreds of thousands of people worldwide every year, males became sicker. More live bacteria were recovered from male mice than from females. Male survival rates were lower. When every variable was controlled except sex, the female immune system simply performed better. In vaccination studies, the differences are equally striking. When men and women receive the same dose of influenza vaccine, women consistently produce higher antibody levels. The difference is so large that some researchers have found women receiving half the standard dose still achieve antibody levels equivalent to men receiving the full dose. This is not because women have a more positive attitude toward vaccination. It is because their immune system mounts a more robust, more aggressive, more effective response, driven by the combination of chromosomal advantage, hormonal modulation, and cellular diversity that defines the female immune architecture. Estrogen enhances immune function through multiple pathways. It increases the expression of toll-like receptors, the molecular sensors on immune cells that detect the presence of bacteria, viruses, and fungi. More toll-like receptors mean faster detection. Faster detection means faster response. Faster response means the infection is contained before it spreads, before it causes systemic damage, before it becomes life-threatening. Estrogen also modulates the balance between pro-inflammatory and anti-inflammatory responses, generally promoting a more effective pathogen-clearing response while limiting the tissue damage that uncontrolled inflammation can cause. Testosterone does the opposite. It is broadly immunosuppressive, it reduces antibody production, it dampens T-cell activity, it lowers the overall vigilance and responsiveness of the immune system. In evolutionary terms, this may have been a trade-off. Testosterone redirected biological resources away from immune defense and toward the muscle mass, bone density, and physical output that males needed to compete and survive in the short term. But the long-term cost of that trade-off is a body that fights infection less effectively, clears pathogens more slowly, builds weaker immunological memory, and dies from infectious disease at higher rates. Now here is where the story must become entirely honest, because the female immune advantage is not a cost-free gift. It is a trade-off, and the cost is real, and millions of women are living with that cost right now. The same immune aggressiveness that protects women from infection makes them more vulnerable to autoimmune disease. 80% of all autoimmune disease patients are women. Lupus, rheumatoid arthritis, multiple sclerosis, Sjögren's syndrome, Hashimoto's thyroiditis, Graves' disease, scleroderma. Type 1 diabetes in adults occurs more frequently in women. Celiac disease occurs more frequently in women. The list of autoimmune conditions that disproportionately affect women is long, and the suffering they cause is profound. Chronic pain, fatigue, organ damage, disability, reduced quality of life. In 2024, researchers at Stanford University identified a specific molecular mechanism that helps explain this connection. The process of X chromosome inactivation, the silencing of one X in every female cell, produces a molecule called ZIST. This molecule and its associated protein complexes coat the inactivated X chromosome. But the immune system can recognize some of these protein complexes as foreign targets, producing auto-antibodies against the body's own molecular components. The very process that gives women their genetic mosaic, their immune diversity, their survival redundancy, also produces a molecular trigger that can set the immune system against itself. Furthermore, the inactive X chromosome in immune cells is not as thoroughly silenced as it is in other cell types. Researchers have found that in lymphocytes and other immune cells, the inactive X can become partially reactivated during inflammatory stress. This means that immune cells in a woman's body can, under certain conditions, express genes from both X chromosomes simultaneously, amplifying the immune response beyond normal levels. This amplification helps fight infection with extraordinary effectiveness. But it can also amplify autoimmune responses, turning the immune fortress's weapons inward. Evolution does not build perfect systems. It builds systems that maximize survival at the population level, even when those systems carry individual costs. The female immune system was optimized for the threat environment that dominated most of human history. A world where infectious disease was the single greatest killer, where a more powerful immune system meant the difference between life and death for a pregnant woman, a nursing mother, a child-carrying caretaker. The cost of that optimization, the risk of autoimmune conditions, particularly in midlife and beyond, was biologically acceptable, because by that point the genes had already been transmitted. There is a cruel irony in how this trade-off was treated by the medical establishment for centuries. Women who reported the symptoms of autoimmune disease, chronic pain, mysterious fatigue, joint inflammation, rashes, cognitive fog, were routinely dismissed. They were told it was anxiety, stress, hysteria, depression. They were told it was in their heads. They were prescribed sedatives and told to rest. What was actually happening in their bodies was the overactivity of the most powerful immune system evolution could build. An immune system so aggressive in its defense that it sometimes turned its weapons on the body it was designed to protect. Understanding autoimmune disease as the cost of the immune fortress does not diminish the suffering. It does not make the pain less real or the disability less devastating, but it gives it a context that is 300,000 years old. It reveals that the system causing the suffering is the same system that has kept women alive through every plague, every epidemic, every pandemic in the history of our species. The same system that cleared the Spanish flu faster, the same system that fought COVID-19 more effectively, the same system that gives female newborns a survival edge in the first hours of life. The immune fortress was not built for comfort. It was built for survival, and the women who live with autoimmune disease are living with the price of that survival, a price that the medical world is only now beginning to understand, acknowledge, and treat with the seriousness it deserves. Chapter 4: The Cellular Clock. Imagine that inside every cell in your body there is a fuse, a physical structure made of DNA. That burns down a little shorter every time that cell divides. When the fuse is long, the cell is young, vigorous, functional, capable of dividing again and again to repair tissue, replace worn-out neighbours, and maintain the organ it belongs to. When the fuse burns down to a critical length, the cell stops. It cannot divide safely anymore. It enters a twilight state, alive but dysfunctional, like a factory that still has its lights on but has stopped producing anything useful, worse than useless actually, because in this twilight state the cell begins releasing inflammatory chemicals, cytokines and other signals that damage the surrounding tissue, accelerating the aging of the entire organ. This fuse is real. It is called a telomere. And the story of telomeres is one of the most important and least discussed chapters in the science of why women outlive men. Telomeres are stretches of repetitive DNA sequences, the letters TTAGGG, repeated hundreds to thousands of times, that cap the ends of every chromosome in every cell. They do not contain genes. They do not code for proteins. Their sole purpose is protection, preventing the chromosome from unraveling, from fusing with other chromosomes, from losing the critical genetic information stored near its ends. Every time a cell divides, the enzymes that copy the DNA cannot quite reach the very end of the chromosome, and a small piece of the telomere is lost. A few dozen base pairs, enough to notice over time. In a young person, telomeres are long, perhaps 10,000 to 15,000 base pairs in the white blood cells commonly used for measurement. By middle age, they have shortened significantly. By old age, many cells have telomeres that are approaching the critical minimum length, the point at which the cell will stop dividing and enter senescence. The accumulation of senescent cells is one of the central drivers of aging. It is not the only one. Aging is multifactorial, involving oxidative damage, protein misfolding, mitochondrial dysfunction, epigenetic drift, and other processes. But telomere shortening is one of the most fundamental, because it directly limits the body's ability to repair and maintain itself. A tissue whose cells can no longer divide is a tissue that cannot heal injuries, cannot replace worn-out cells, thus cannot maintain its function over time. This is why aging affects everything. The heart weakens, the blood vessels stiffen, the skin thins, the immune system slows, the brain loses volume, the muscles waste, the bones thin, the organs gradually fail. The cellular fuses are burning out, and the body's capacity for self-repair is declining with them, and women start with longer fuses. This is not a metaphor. It is a measurable, reproducible, confirmed biological fact. Multiple studies, using multiple measurement techniques, quantitative fluorescence in situ hybridization, flowfish, telomere restriction fragment analysis, have demonstrated that female telomeres are longer than male telomeres from birth. The 2022 study in Aging Cell was not the first to show this. A 2014 meta-analysis pooling data from multiple studies confirmed the same finding. A 2006 study analysing umbilical cord blood found that female newborns had longer telomeres than male newborns. The difference is consistent, reproducible, and present before any environmental or behavioural factor has had time to exert an influence. The difference at birth is between 100 and 300 base pairs. That may sound small in absolute terms, but in a system where each cell division costs approximately 50 to 100 base pairs, an extra 200 base pairs represents 2 to 4 additional cell divisions before senescence. Multiply that across billions of cells, across dozens of tissues, across decades of life, and the cumulative effect is substantial. It is the difference between a body that maintains its repair capacity into the 70s and a body whose repair capacity begins to fail in the 60s. It is the difference between organs that hold their function for 7 decades and organs that begin to fail after 6. And the advantage does not end at birth. It grows. Estrogen, as we have already seen, activates telomerase, the enzyme that can rebuild telomeres by adding new TTAGGG repeats to their ends. Telomerase is the body's counteragent to the telomere clock, the enzyme that can slow, and in some cells partially reverse, the shortening process, and estrogen turns it on, continuously, for roughly 35 to 40 years. Research published in multiple journals has confirmed that estrogen upregulates telomerase activity through direct interaction with the telomerase reverse transcriptase gene promoter. Estrogen literally turns on the gene that makes the enzyme that maintains the fuses. It does not do this sporadically or occasionally. It does this as a regular, ongoing function of the hormonal environment of the female body throughout the reproductive years. Men have no equivalent hormonal mechanism for telomere maintenance. Testosterone does not activate telomerase. If anything, the higher metabolic demands imposed by testosterone, the larger muscle mass requiring more cell divisions for maintenance, the higher metabolic rate generating more oxidative stress, accelerate telomere attrition, and our male cells are losing their fuses faster while receiving no hormonal help to repair them. The result is a divergence that widens with every passing year. At birth, the female telomere lead is modest, perhaps 200 base pairs. By early adulthood, the hormonal amplification of the difference has begun. By middle age, the gap has compounded. By old age, the difference in biological age between a man and a woman of the same chronological age is measurable, and it is consistently in the woman's favour. This is one of the reasons why the sex difference in mortality is not uniform across the lifespan, but follows a predictable curve. In childhood, the gap is present but relatively small, driven primarily by the chromosomal advantage and the intrinsic differences in infant robustness. In adolescence and early adulthood, the gap begins to widen as hormonal differences take hold. Estrogen begins its protective work in women. Testosterone begins its expensive acceleration in men. In midlife, the gap is at its widest. Decades of divergent hormonal environments have produced measurably different rates of cellular aging, cardiovascular degeneration, immune function decline, and organ deterioration. After menopause, the gap narrows slightly. Estrogen's protective effects diminish, and women's telomere maintenance slows. But the accumulated lead of decades cannot be erased. The capital built during the years of estrogen protection persists. There is an emotional dimension to this that deserves acknowledgement. Every woman listening to this who is past menopause, or approaching it, or in the thick of it, every woman who has felt the changes that come when estrogen withdraws, the shifts in skin and sleep and energy and joint comfort and cognitive sharpness, and the 100 other adjustments the body makes, may hear this information and feel a sense of loss. The shield is lowering. The telomerase activation is declining. The cellular clocks are no longer being maintained at the same rate. That feeling is real, and it deserves to be honoured. But what is also real, and what is rarely said, is that by the time menopause arrives, the estrogen shield has already done its work. 35 to 40 years of cardiovascular protection, of telomere maintenance, of anti-inflammatory defense, of cellular repair, that is not erased when estrogen declines. The fortress was built. The walls are still standing. The male body, Naswar, at the same age never had those walls to begin with. It was running without the shield from the start. You are not unprotected. You are carrying the accumulated capital of decades of maintenance that no man your age possesses. The shield may have lowered, but the fortress endures. Chapter 5. Mother's Curse. There is a structure inside every cell of your body that was not always yours. It was not always human. It was not always part of any animal. Over 1 billion years ago, long before the first fish, the first land plant, the first insect, it was a free-living bacterium, swimming through ancient oceans, making its own way in the world. And then, in one of the most consequential events in the entire history of life on Earth, an ancestor of your cells swallowed it. The bacterium did not die, it survived inside its host. Over millions of years, a partnership developed. The host cell provided protection and nutrients, and the bacterium provided something the host desperately needed: energy. Specifically, the bacterium was extraordinarily efficient at converting nutrients into adenosine triphosphate, ATP, the universal energy currency of cellular life. The host cell that harboured this energy-producing partner had a tremendous advantage over cells that did not. It could power larger bodies, more complex functions, more demanding lifestyles. It could do more. It could become more. That ancient bacterial partner is now called the mitochondrion, and you have hundreds to thousands of them in nearly every cell of your body. They produce approximately 90% of the energy your body uses. Every heartbeat, every thought, every breath, every step, yes, every blink, every nerve impulse, every muscle contraction, all of it powered by the descendants of a bacterium that was swallowed over 1 billion years ago. But here is the fact that changes everything about male and female longevity. Mitochondria have their own DNA, a small circular genome separate from the chromosomal DNA in the cell's nucleus. And this mitochondrial DNA is inherited exclusively from your mother. When a sperm cell fuses with an egg, the egg's cellular machinery systematically identifies and destroys the sperm's mitochondria. The father contributes half of the nuclear genome, half of the chromosomal blueprint, but zero mitochondria. Every single mitochondrion in your body, in every cell, in every organ, traces its lineage back through an unbroken maternal line. Your mitochondria came from your mother. Hers came from her mother. Hers came from hers. Back and back and back, mother to daughter, for hundreds of thousands of years in the human lineage, and for hundreds of millions of years in the mammalian lineage before that. This purely maternal inheritance has a consequence that most people have never heard of. A consequence so strange, so counterintuitive, and so deeply relevant to the question of why women outlive men, that evolutionary biologists gave it a name borrowed from fairy tales' mother's curse. The logic unfolds like this. Because mitochondrial DNA passes only through females, natural selection can only act on mitochondrial mutations as they affect female fitness. If a mutation arises in the mitochondrial genome that harms females, reducing their energy production, damaging their cells, lowering their fertility, shortening their lives, that mutation will be filtered out. Females carrying it will be less likely to survive and reproduce. Their mitochondrial lineage will die out. The mutation will be removed from the population. The system works for females, but if a mutation arises in the mitochondrial genome that harms only males, reducing male fertility, damaging male-specific tissues, accelerating male aging, natural selection is completely blind to it. The mutation is passed through mothers. The mothers are unaffected, they survive, they reproduce. They pass the mutation to their sons and daughters. The daughters are unaffected, they survive. They pass it to their sons and daughters. The mutation drifts through the population indefinitely, expressing its harm only in males, invisible to the evolutionary filter that would remove it if it harmed females. This means that the mitochondria inside every man's body are, to some measurable degree, optimized for the female body. They have been shaped by hundreds of millions of years of selection that tested their performance exclusively in women. Mutations that harm men but not women have been allowed to accumulate. The result is a subtle but persistent mismatch between male physiology and mitochondrial function, a mismatch that does not exist for women. The mother's curse hypothesis was proposed theoretically before it was proven empirically. But in 2012, researchers at Monash University in Australia published the first large-scale experimental evidence, testing mitochondrial genomes from different populations of fruit flies. They found that certain mitochondrial variants significantly reduced male lifespan and male fertility, while leaving females completely unaffected. The mutations targeted male reproductive function most dramatically, but they also affected broader aspects of male vitality and aging. Then, in 2016, the evidence became even more specific. A research team at the Fred Hutchinson Cancer Research Center in Seattle identified the first true mother's curse mutation in animals, working with fruit flies, an experimental undertaking so labour-intensive that it required 1.5 years, hundreds of thousands of individual flies, and a team of dedicated technicians. They found a single mutation in a mitochondrial gene coding for a critical component of the energy production machinery. The mutation reduced the activity of that component by approximately 20%. In female flies, this 20% reduction had zero detectable consequences. No change in lifespan, no change in fertility, no change in health, nothing. In male flies, the same mutation, the same gene, the same 20% reduction, rendered them completely sterile. Their sperm was, in the researcher's description, a tangled mess. The same mutation, no effect on females, devastating to males. Males represent an evolutionary dead end for mitochondria, said Dr. Maulik Patel, one of the lead researchers. The statement is not an opinion or a provocation. It is a description of an evolutionary reality. Mitochondria pass through females. Males are a dead-end branch. Mitochondria have no evolutionary incentive to protect male function, or because male function does not affect mitochondrial transmission. The research team then tested whether the nuclear genome, the chromosomal DNA inherited from both parents, had evolved compensating mechanisms. When they introduced the sterility-causing mitochondrial mutation, into 12 different genetic backgrounds of flies. 8 of the 12 backgrounds completely suppressed the sterility effect. The nuclear genome had evolved countermeasures, but the countermeasures were incomplete. Only 8 of 12 backgrounds were protected. The other 4 remained vulnerable. This is the arms race at the heart of Mother's curse. The mitochondria accumulate mutations that harm males. The nuclear genome evolves suppressors to compensate, but the suppressors are never perfect, never complete, never absolute. Some male-harming mutations slip through. Some genetic backgrounds are less protected than others. The result is a persistent, low-grade disadvantage for male biology, a tax on male survival that females never pay. The implications for human health are potentially enormous. Between 7 and 10% of men have some form of fertility defect, either complete sterility or significantly reduced fertility, and for the majority of these cases, the genetic cause is unknown. Mitochondrial mutations have already been implicated in some cases of human male infertility, and the Mother's Curse framework suggests that there may be many more such mutations quietly affecting male reproductive and general health that have never been identified, because they cause no symptoms in the mothers and daughters who carry them, and because medical research, until very recently, has not been systematically looking for sex-specific mitochondrial effects. Some researchers have argued that Mother's Curse may contribute not just to male infertility, but to the overall male lifespan disadvantage. If the mitochondria, the organelles responsible for 90% of cellular energy production, are running slightly less efficiently in males, then every energy-demanding process in the male body is marginally compromised. Cellular repair is marginally less efficient. Brain function is marginally more metabolically stressed. Cardiac output is marginally more costly. Muscle maintenance is marginally more expensive. Over decades, these marginal inefficiencies compound into measurable differences in organ health, cellular damage accumulation, and ultimately years of life. For women, none of this applies. Your mitochondria are running on hardware that was quality tested in your sex, in your hormonal environment, in your metabolic context, for hundreds of millions of years. Every generation of selection that refined your mitochondrial genome was testing it in a female body. Every harmful variant that appeared was filtered out, because it affected females, and selection could see it. The energy that powers every cell in your body right now, every heartbeat, every thought, every breath, is being produced by organelles that were optimized for you, not for a generic human, for you, for a female body. The man beside you carries the same organelles, but his were optimized for someone else's body. And in the difference between hardware optimized for its host and hardware optimized for a different host, there is a margin, small, quiet, persistent, that compounds over a lifetime into years. Chapter 6. Built for famine. In November of 1846, the first heavy snows fell on Truckee Lake in the Sierra Nevada, sealing the fate of 87 American emigrants who had taken a disastrous shortcut on their way to California. They would become known as the Donner Party. They would become one of the most infamous survival stories in American history, remembered for the starvation, the desperation, the cannibalism that consumed the group as winter stretched from weeks into months with no rescue and no food. But within the horror, there is a pattern that has been analysed by anthropologists, published in peer-reviewed journals, and confirmed by demographic data from catastrophes spanning 3 centuries and 4 continents. A pattern that has everything to do with the question at the heart of this documentary. The men died at nearly twice the rate of the women. 30 out of 53 men perished. 10 out of 34 women died. Male mortality, 57%. Female mortality, 29%. Women survived at nearly double the rate of men, under identical conditions of extreme cold and starvation. When anthropologist Donald Grayson analysed the mortality data from the Donner Party in a study published in the Journal of Anthropological Research, he controlled for age. He accounted for the fact that survivors tended to be younger than those who died. But even after adjusting for age, the sex difference held. It was not that more young women happened to survive. It was that, across age categories, women were more resistant to the combination of cold and starvation than men. Women are simply better fit biologically to withstand cold and famine than men, Grayson concluded. They are smaller, with 17% less body area to volume ratio, which means they conserve heat more efficiently, and they carry proportionally more body fat. The Donner Party is a dramatic case, but it is not an anomaly. It is a single data point in a vast and consistent pattern. During the Irish potato famine of 1845 to 1849, women outlived men by nearly 4 years, 22.4 years versus 18.7 years, even as 1 million people starved to death and another 1 million fled the country. During the Ukrainian Holodomor of 1933, when Joseph Stalin's policies deliberately starved millions of Ukrainians, women's life expectancy was almost 50% higher than men's, 10.85 years versus 7.3 years. During the Swedish famine of 1772 to 1773, women outlived men by 1.64 years. During the Icelandic measles epidemics of 1846 and 1882, women outlived men by approximately 1 to 2 years. The pattern spans centuries, continents, and cultures, and in every case, the largest single contributor to the female survival advantage was not adult resilience, it was infant survival. Newborn girls survived these catastrophes at higher rates than newborn boys. In some populations, the 0 to 1 age group accounted for over 70% of the total survival gap between the sexes. Why? What is it about the female body that makes it more resistant to starvation, cold, and disease, even in infancy, even before any behavioural or lifestyle factor has had time to exert an influence? The answer lies in what the female body carries, how it stores energy, and how it uses that energy when external supplies fail. Women carry a higher percentage of body fat than men. This is not a marginal difference. The average healthy young woman's body is approximately 20 to 25% fat. The average healthy young man's body is approximately 15 to 20% fat. The difference is not cultural. It is not the result of diet or exercise habits. It is hormonally driven, genetically programmed, and present across every human population ever studied. Estrogen promotes fat storage. Testosterone promotes lean muscle tissue. The hormonal environments of the male and female bodies literally build different physical compositions from the same raw materials, and the fat itself is different. Women's fat is preferentially stored subcutaneously, beneath the skin, distributed across the hips, thighs, breasts, and buttocks. Men's fat is more likely to be stored viscerally, around the organs, inside the abdominal cavity. This difference in distribution is not cosmetic. It is medically profound. Subcutaneous fat is metabolically safe. It is relatively inert, does not interfere with organ function, produces lower levels of inflammatory cytokines, and is readily mobilized for energy during periods of caloric deficit. It is, in essence, a clean fuel reserve, easily accessible, efficiently burned, and stored in a location that does not damage the surrounding tissue. Visceral fat is metabolically dangerous. It is associated with higher levels of systemic inflammation, insulin resistance, liver disease, cardiovascular disease, and type 2 diabetes. It interferes with organ function. It produces inflammatory signals that accelerate aging throughout the body. And it is less efficiently mobilized for emergency energy, meaning that in a famine scenario, visceral fat is a less effective survival reserve than subcutaneous fat. Estrogen promotes subcutaneous storage. Testosterone promotes visceral storage. The same hormonal system that protects women's hearts, maintains their telomeres, and powers their immune systems also ensures that their energy reserves are stored in the safest, most survival-optimized configuration available. The female body's fat distribution is not a flaw, it is a survival system. The body you were taught to criticize in the mirror is the body that survived every famine in human history. The metabolic advantage extends beyond fat composition and distribution. Women's basal metabolic rate is lower than men's, proportional to their smaller average body size and lower lean muscle mass. In normal times, this means women need fewer calories to function. In a famine, this is a decisive survival advantage. A body that needs less fuel can survive longer on the same reserves. A woman's smaller body and lower metabolic rate mean that her fat reserves, which are already proportionally larger than a man's, last longer. Men's larger muscle mass is a survival advantage in times of plenty. It provides strength for physical labour, hunting, defence. But muscle is metabolically expensive. It burns calories even at rest. In a famine, that expensive tissue becomes a liability. The body begins to catabolize muscle for energy, and the process of breaking down muscle tissue generates toxic byproducts, ammonia, urea, that stress the kidneys and liver. Men, with more muscle mass to maintain and more metabolic demand to meet, burn through their reserves faster. Their organs are stressed harder. They die sooner. There is evidence that women's bodies are also more efficient at the metabolic transition from glucose burning to fat burning during prolonged fasting. When food intake drops dramatically, the body must shift from using dietary glucose as its primary fuel source to using ketone bodies derived from stored fat. This transition, called ketogenesis, is metabolically complex and can cause significant disruption, particularly in its early stages. Well, some research suggests that the female body, primed by the metabolic flexibility needed for pregnancy and lactation, which require dramatic shifts in fuel utilization, manages this transition more smoothly. Consider what this means in evolutionary terms. For 300,000 years, the female body was the body that had to survive famine while pregnant, while nursing, while carrying a dependent child. The metabolic demands of pregnancy, building a placenta, growing a fetus, maintaining blood volume, preparing for lactation, are enormous. The metabolic demands of lactation are even greater. A breastfeeding woman may need 500 additional calories per day. These demands had to be met in an environment where caloric scarcity was the norm, not the exception. A body that could not store energy efficiently, that could not access that energy under stress, could not maintain its critical functions when food was scarce, that body would lose the pregnancy, lose the child, lose its genetic legacy. Evolution built the female body to eat when food was available and endure when it was not. Every mechanism we have discussed, the higher fat percentage, the subcutaneous distribution, the lower metabolic rate, the metabolic flexibility, the estrogen-mediated hormonal regulation of energy storage, is a component of this survival system. A system that was tested generation after generation across ice ages and droughts and famines and seasonal scarcities that lasted hundreds of thousands of years. The fat that modern culture teaches women to despise is, from an evolutionary perspective, one of the most critical survival adaptations in the human species. It kept mothers alive through famine. It sustained pregnancies during scarcity. It powered lactation when foraging failed. It kept grandmothers productive when younger adults were weakened. Every woman who has ever looked in a mirror and wished her body carried less fat is looking at a survival system that was refined over 300 millennia to do exactly one thing: keep her alive when everything around her was dying. And it works. The Donner Party proved it. The Irish famine proved it. The Ukrainian Holodomor proved it. The Swedish famines, the Icelandic epidemics, the freed slave populations of Liberia all proved it. Under the most extreme conditions humanity has ever endured, the female body's energy storage and metabolic efficiency system delivers exactly what it was built to deliver: survival. The male body, built by testosterone for physical performance and short-term competitive advantage, pays the price of that design when conditions turn harsh. The strength that wins fights does not win famines. The muscle mass that impresses mates becomes the liability that kills. The metabolic engine that burns hot in good times burns through its fuel in bad times. The female body was not built for good times. It was built for the worst times, and in the worst times, it endures. Chapter 7. The Grandmother Revolution. Here is one of the strangest facts in all of biology. A fact so unusual, so seemingly illogical, so contrary to the basic principles of how evolution works, that it puzzled scientists for decades before anyone could explain it. A fact that, once you understand it, transforms not just how you think about aging and menopause, but how you think about the entire structure of human civilization, the evolution of human intelligence, and the role of older women in the survival of the species. Human females live for decades after they stop reproducing. Step back from this fact for a moment and consider how extraordinary it is. Evolution is driven by reproduction. That is its engine, its logic, its only currency. Genes that help an organism survive and reproduce are passed on. Genes that don't are lost. The body is, in a brutal sense, a vehicle for the transmission of genes. And once the body can no longer transmit genes, once it can no longer reproduce, it has, in strict evolutionary terms, fulfilled its purpose. In the vast majority of animal species, this logic plays out straightforwardly. Females remain fertile until close to the end of their lives. A female elephant seal, a female bear, a female chimpanzee, each continues to reproduce until her body cannot sustain the effort anymore, and death follows relatively soon after. Reproduction and death are tightly coupled. There is no extended post-reproductive lifespan. There is no biological investment in keeping the body alive and functional once reproduction has ended. But human women defy this pattern in a way that has no close parallel in the natural world. The average age of menopause is approximately 51. The average female life expectancy in developed countries is now in the mid-80s. That means women live, on average, more than 30 years after their fertility ends. 3 decades. In some women, 4 decades. An entire adult lifetime spent in a body that is no longer reproducing but that is still being maintained, still being repaired, still being kept alive by every system we have discussed. The chromosomes, the hormones, the immune cells, the telomeres. Why? If evolution rewards reproduction, why would it build a body that invests such enormous biological resources in survival decades past the reproductive endpoint? Why pour energy into maintaining a postmenopausal woman's heart and brain and bones and immune system when she can no longer pass her genes directly to the next generation? The traditional answer, the one that dominated medical and popular thinking for most of the 20th century, was that postmenopausal longevity is an accident, a byproduct of modern medicine and improved living conditions. Humans used to die young, the argument went, and menopause was simply a failure point that most women in ancient times never reached. Postmenopausal life is a modern phenomenon, not an evolved adaptation. This answer is wrong. Demographic data from modern hunter-gatherer populations, the populations that most closely approximate the living conditions of our ancestors for most of human history, shows that roughly 1/3 of adult women at any given time are postmenopausal. Women in these populations regularly live into their 60s and 70s. Postmenopausal life is not a modern invention. It is an ancient, consistent feature of human demography that has been present for tens of thousands of years at minimum, and likely for much longer. The answer that finally made sense of this puzzle came not from a laboratory, but from the field, from an anthropologist watching grandmothers dig tubers in the Tanzanian sun. In the late 1990s, Kristen Hawkes and her colleagues at the University of Utah were studying the Hadza people of Tanzania, one of the last remaining hunter-gatherer populations on Earth. The Hadza live much as our ancestors did for most of human history, foraging for wild foods, hunting game, living in small bands, raising children without agriculture, without domesticated animals, without any of the modern infrastructure that separates most of us from the conditions that shaped our species. Hawkes observed something that changed evolutionary biology. Postmenopausal Hadza women were not idle. They were not marginal. They were not being passively cared for by their families. They were among the hardest working and most productive members of the group. Grandmothers spent long hours digging tubers, underground storage organs of wild plants, thus rich in calories, but deeply embedded in hard soil and requiring significant physical effort and adult skill to excavate and process. Young children could not effectively dig tubers. Their arms were not strong enough, their hands not skilled enough, their understanding of where to dig and how to process the tough, fibrous roots not developed enough. Mothers with nursing infants could not dedicate sufficient time to tuber harvesting, while also caring for their babies and managing older children. But grandmothers could, and they did. The effect was transformative. When grandmothers provided food for their grandchildren, mothers could wean their current children earlier and become pregnant again sooner. Grandmothers were, in effect, subsidizing their daughters' reproduction, allowing them to have more children, more frequently, with higher child survival rates. Nick Blurton-Jones's comprehensive 2016 demographic study of the Hadza confirmed the effect with data. Living grandmothers had strong, measurable positive effects on grandchild survival. Children with living, active grandmothers were more likely to survive to adulthood than children without them. This created a powerful selection pressure, one of the most elegant and consequential in human evolution. Women who lived longer after menopause had more grandchildren who survived. Their genes, including the genes that promoted longer post-reproductive lifespan, were passed on at higher rates. Over thousands of generations, this pressure gradually extended female lifespan further and further beyond the reproductive window. Computer simulations conducted by Peter Kim and colleagues confirmed that grandmothering alone, without any other selective pressure, could drive the evolution of human-like longevity from a chimpanzee-like starting point. In chimpanzees, few females survive past their reproductive years. The simulations showed that the introduction of grandmothering as a factor was sufficient to produce a population in which approximately 1/3 of adult females were postmenopausal, matching the pattern observed in actual human hunter-gatherer populations. The grandmother hypothesis, as it came to be known, reframes everything about menopause and postmenopausal life. Menopause is not a failure. It is not a breakdown. It is not the end of a woman's biological purpose. It is a strategic shift, a redirection of biological resources from direct reproduction, which becomes increasingly risky and costly with age, to indirect reproduction through the survival of grandchildren. The decades of postmenopausal life are not surplus time that evolution forgot to remove. They are the direct result of selection favouring women who lived longer, because those women's grandchildren survived at higher rates, and their genes, the genes for longevity, proliferated. The idea was first hinted at in 1957, when evolutionary biologist G.C. Williams proposed that menopause might be an adaptation. Williams argued that at some point in the aging process, the risk of continued reproduction, the danger of complications, birth defects, maternal death, exceeded the benefit of producing one more child. A woman who stopped reproducing and instead invested her remaining energy in her existing children and grandchildren would, over time, leave more surviving descendants than a woman who continued to reproduce until the effort killed her. The grandmother hypothesis builds on Williams's insight and extends it into a full evolutionary model, and its implications reach far beyond demography. If grandmothering drove the evolution of extended female lifespan, then grandmothers are not peripheral to human evolution. They are central to it. Grandmothers may have been the selective pressure that made humans long-lived in the first place. The evidence extends beyond the Hadza. In pre-industrial Finland, studies of parish records showed that the presence of a living maternal grandmother significantly increased grandchild survival rates. In Gambia, as similar patterns were observed, in historical Japanese populations, in Canadian historical populations, in population after population where the data existed to be analysed, grandmothers mattered. Their presence correlated with better outcomes for the children around them, and their contribution went beyond food provision. Grandmothers transmitted knowledge, which plants are safe to eat and which are poisonous, how to prepare food to remove toxins, how to treat common illnesses with available plants, when the rains will come, where to find water in a drought, what the signs of an approaching storm look like, how to care for a sick child, how to deliver a baby, how to manage social conflicts within the group. Whereas in populations without written language, which means all human populations for the vast majority of our species' existence, the grandmother was the primary repository of accumulated knowledge. She had lived through things no younger member of the group had experienced. She remembered the famine of 30 years ago. She remembered the flood. She remembered the epidemic. She knew what had worked and what had failed. She was a living library, an archive of survival information accumulated across decades of observation and experience. No society at any point in 300,000 years of human history could afford to lose that knowledge, and evolution responded by building bodies that preserved it, bodies that maintained cognitive function, physical capability and social engagement for decades past the reproductive endpoint. The grandmother is not a relic. She's not a remnant. She's not a burden on a society optimized for youth. She's a technology, a living, breathing, irreplaceable technology of knowledge preservation and transmission that evolution spent hundreds of thousands of years perfecting. Every woman past menopause who has ever been made to feel that her best years are behind her, that she is past her usefulness, that she has nothing left to contribute. Every woman who has internalized the message that youth is the only form of female value, is living inside a body that evolution specifically, deliberately, measurably, built to be useful beyond reproduction. A body whose post-reproductive survival was so valuable to the species that natural selection invested enormous biological resources in maintaining it. You were not supposed to fade. You were supposed to stay. And the fact that you are still here, still sharp, still capable, still carrying knowledge and judgment and emotional intelligence that no algorithm can replicate, is not an accident of modern medicine. It is the fulfillment of an evolutionary design 300,000 years in the making. Chapter 8. The Stress Advantage. For most of the 20th century, the science of stress operated on an assumption so deeply embedded that most researchers never thought to question it. The assumption was that the stress response was universal, that all human bodies, male and female, responded to threat in the same way. The model was called fight or flight. It was first articulated by physiologist Walter Cannon in the 1920s, based on research conducted primarily on male animals. The model described a cascade of physiological changes triggered by the perception of danger. The adrenal glands flood the bloodstream with adrenaline and cortisol. The heart rate increases, blood pressure rises, blood is redirected from the digestive and immune systems to the skeletal muscles, pain sensitivity decreases, and the body is prepared for one of two responses: fight the threat or flee from it. Fight or flight became one of the most famous concepts in all of biology. It was taught in every introductory psychology and physiology course. It was referenced in self-help books, corporate wellness programs, military training manuals, and popular science articles. It was treated as the human stress response. In the year 2000, a psychologist at UCLA named Shelley Taylor read the literature and noticed something that should have been obvious. The vast majority of the research on which the fight-or-flight model was based had been conducted on males, male rats, male mice, male human subjects. The female stress response had simply been assumed to be the same. It had not been independently studied with anything approaching the depth or rigour applied to males. Taylor and her colleagues published a paper that should have detonated like a bomb in the field. Biobehavioral responses to stress in females: tend and befriend, not fight or flight. The paper pointed out something that was, once stated, breathtakingly obvious. The fight or flight model makes evolutionary sense for males, but it makes very little sense for females, particularly females who are pregnant, nursing, or caring for dependent young. A pregnant woman cannot fight a predator with any reasonable chance of success. Her body is carrying another body. She's slower, she's less agile, she's more vulnerable to injury, and injury does not just threaten her life. It threatens the life of the fetus she is carrying, the investment of months of biological resources. A nursing mother cannot flee at full speed. A woman carrying an infant on her hip cannot outrun a predator. A woman surrounded by children too young to run on their own cannot abandon them to save herself, not without catastrophic genetic consequences, because those children carry her genes. For the female of virtually every mammalian species, fight or flight is not a first-line strategy, it is a last resort. An emergency response for situations where tending and befriending has failed. Tend and befriend. This is the female stress response, and it is fundamentally different from the male response in its neurochemistry, its behavioral expression, and its long-term health consequences. Under stress, Taylor proposed, females are more likely to tend, to engage in nurturing behaviors that protect their offspring and reduce the vulnerability of the group. They check on the children, they feed the children, they pull the children closer, they assess the environment for threats and take protective action, not by fighting or fleeing, but by sheltering, soothing, and securing. And females are more likely to befriend, to seek out social alliances, to strengthen relationships, to create networks of mutual aid and shared vigilance. When a female animal is stressed, she moves toward the group, not away from it. She seeks out other females, she grooms them, she sits close to them, she engages in behaviors that reinforce social bonds, bonds that provide safety in numbers, shared childcare, cooperative defense, and access to resources that no individual could secure alone. The neurochemical basis for this difference centers on oxytocin, sometimes called the bonding hormone, sometimes the love hormone, though these popular names undersell its biological significance. Oxytocin is released under stress in both males and females, but in females, its effects are amplified by estrogen. Oxytocin promotes trust, empathy, social affiliation, and nurturing behavior. It reduces the cortisol response. It lowers blood pressure. It dampens the inflammatory cascade that stress hormones trigger. It is, in a very real sense, an anti-stress hormone, a chemical that counteracts the damage of the fight-or-flight response. In males, testosterone inhibits oxytocin's calming and bonding effects. Instead of promoting social connection, the male hormonal cocktail under stress tips toward aggression, territorial behavior, and isolated coping. The male stress response is more likely to produce confrontation or withdrawal, both of which are metabolically expensive and in the long term physiologically damaging. The health consequences of this difference are enormous. The fight or flight response is metabolically expensive, chronic activation of the stress response, chronic elevation of cortisol, chronic increases in blood pressure, chronic suppression of the immune system. And chronic inflammation is one of the most well-documented contributors to cardiovascular disease, immune dysfunction, cognitive decline, metabolic syndrome, and premature death. Researchers call this accumulated physiological wear and tear allostatic load, and decades of research have shown that higher allostatic load predicts earlier death. Men's stress response, shaped by testosterone, predisposes them to chronic fight-or-flight activation. When male animals are subjected to chronic stress, they show more sustained cortisol elevation, more persistent inflammatory markers, more cardiovascular damage. When male humans experience chronic stress, the stress of poverty, unemployment, discrimination, loneliness, grief, they are more likely to cope through withdrawal, substance use, aggression, or stoic endurance that keeps the cortisol flowing without relief. These coping strategies are not just culturally determined, they are hormonally facilitated. Testosterone pushes the male stress response toward isolation and confrontation. Women's stress response, shaped by estrogen and oxytocin, predisposes them to social buffering, the reduction of stress through connection, mutual support, and communal coping. When a woman is stressed, she is more likely, not because she's socialized to, but because her neurochemistry pushes her to call a friend, to talk to her sister, to sit with her mother, to seek out the company of other women. And that connection is not just emotionally comforting, it is physiologically protective. Social contact triggers additional oxytocin release, which further reduces cortisol, further lowers blood pressure, further dampens inflammation. The relationship between social connection and longevity is one of the most robust findings in all of public health research. A meta-analysis published in PLOS Medicine found that social isolation increases mortality risk by approximately 30%, a risk comparable to smoking 15 cigarettes a day. Loneliness is not just emotionally painful, it is physiologically lethal. And across cultures, across age groups, across every population studied, women maintain larger and deeper social networks than men. They have more close friendships, they engage in more emotional disclosure, they provide and receive more social support, they are more likely to reach out when they are struggling, they are more likely to accept help when it is offered. These patterns are often attributed to socialization. Women are taught to be more relational, more emotionally open, more nurturing. And socialization plays a role. But the Tend and Befriend research suggests that the foundation is biological. The female stress response is neurochemically oriented towards social connection because for hundreds of thousands of years, social connection was the survival strategy that worked for pregnant, nursing, child-carrying females. The woman who fought died. The woman who fled lost her children. The woman who gathered allies, who tended her young and befriended her community, survived. And her daughters inherited the neurochemistry that made that strategy instinctive. The survival advantage compounds over a lifetime. Every time a woman responds to stress with connection rather than isolation, she triggers a physiological cascade that reduces the biological damage of chronic stress. Her cortisol drops, her blood pressure stabilizes, her inflammation decreases, her immune system maintains function, her telomeres shorten more slowly. Every time a man responds to stress with withdrawal, aggression, or stoic endurance, his cortisol stays elevated. His blood pressure remains high, his inflammation persists, his immune system is suppressed, his telomeres shorten faster. The difference is not about character, it is about chemistry. The female stress response is not gentler because women are weaker. It is more socially oriented because evolution built it that way. Because for a female mammal, the survival strategy that preserved her life and her offspring's life was not combat or escape. It was community, it was tending, it was befriending, and 300,000 years of that strategy have woven it into the neurochemistry of the female brain, making it as fundamental to the female body as the XX chromosomes, the estrogen shield, and the immune fortress. Connection is not a luxury, it is a survival mechanism. And the women who instinctively reach for it, who call their daughters, who meet their friends for coffee, who sit with their sisters in the hard moments, who gather in groups to process grief and celebrate joy, are not being sentimental. They are enacting a 300,000-year-old survival protocol that their bodies were specifically built to execute. Chapter 9. The Iron Paradox and the Hidden Logic of Menstruation. There is a process that happens every month in the bodies of roughly 1.5 billion women worldwide. a process that has been called a curse by dozens of cultures across thousands of years, a process that has been used to justify excluding women from temples, from sacred spaces, from positions of authority, from public life, from kitchens, from sleeping beside their husbands. A process that is still, in the modern world, a source of stigma, shame, and silence, marketed as something to hide, discussed in whispers, treated as an inconvenience to be endured and minimized. Menstruation, the shedding of the uterine lining, the monthly bleed. What if the curse is actually a cleaning? What if the inconvenience is actually a maintenance protocol? What if the very process that has been used for millennia to mark women as impure is, in biological reality, one of the mechanisms that keeps them alive? The connection lies in iron. And the story of iron in the human body is one of the most elegant and least appreciated chapters in the science of female longevity. Iron is essential for life. It is the atom at the centre of hemoglobin, the molecule in red blood cells that carries oxygen from the lungs to every tissue in the body. Without iron, your blood cannot carry oxygen. Without oxygen, your cells cannot produce energy. Without energy, you die. Iron is fundamental, but iron is also dangerous. In excess, iron is a potent oxidant, a catalyst for the production of reactive oxygen species, the free radicals that damage DNA, destroy proteins, rupture cell membranes, and accelerate the aging of every tissue they touch. Iron does not just sit passively in the body when it accumulates beyond what is needed. It actively participates in the chemistry of destruction, driving the Fenton reaction, in which iron ions react with hydrogen peroxide to produce hydroxyl radicals, one of the most damaging molecules in all of biochemistry. And the human body has very limited mechanisms for getting rid of excess iron. Unlike most other minerals, there is no excretory pathway for iron. The body cannot dump excess iron in urine the way it dumps excess sodium or potassium. Once iron is absorbed from the diet, it stays. It accumulates. Slowly, year after year, it builds up in the liver, the heart, the pancreas, the brain. This accumulation is associated with increased risk of heart disease, liver disease, diabetes, a neurodegenerative disease, and cancer. Iron overload is a slow, quiet, cumulative form of oxidative damage that progresses relentlessly over a lifetime. Men have no regular mechanism for iron loss. They absorb iron from their diet. They retain it. It accumulates. By middle age, the average man's serum ferritin levels, the standard laboratory measure of iron stores, are typically 2 to 3 times higher than those of a premenopausal woman. This excess iron contributes to oxidative stress, vascular inflammation, and the accelerated aging of multiple organ systems. Some researchers have identified elevated iron stores as an independent risk factor for cardiovascular disease, a risk factor that operates alongside and compounds the effects of cholesterol, blood pressure, smoking, and diabetes. Women through menstruation lose iron every month, approximately 30 to 40 milligrams per cycle on average. This loss has been traditionally framed as a problem. Iron deficiency anemia, the need for iron supplementation, the fatigue and weakness that can accompany heavy periods, and for some women, particularly those with heavy menstrual bleeding or inadequate dietary iron intake, iron deficiency is a genuine health concern that deserves serious medical attention. But at the population level, the monthly iron loss of menstruation keeps women's iron stores in a range that is, from a cardiovascular and oxidative damage perspective, significantly protective. Premenopausal women's iron levels sit in a sweet spot, high enough to maintain adequate oxygen carrying capacity, low enough to avoid the oxidative damage of excess accumulation. This sweet spot is maintained automatically, monthly, by the same process that has been called a curse for 3,000 years. The evidence for this protective effect is multi-layered. Premenopausal women have dramatically lower rates of heart disease than men of the same age. After menopause, when menstruation stops and iron stores begin to rise toward male levels, women's cardiovascular risk increases sharply. The timing of this increase aligns not only with the decline in estrogen, but with the cessation of monthly iron loss. Some researchers have argued that the postmenopausal rise in cardiovascular disease is not entirely an estrogen withdrawal effect, that the concurrent rise in iron stores plays an independent and significant role. Studies of blood donors provide supporting evidence. It says men who donate blood regularly, effectively mimicking the periodic iron loss of menstruation, have been found in some studies to have lower rates of cardiovascular disease. The mechanism is straightforward. Removing blood removes iron. Lower iron means less oxidative damage. Less oxidative damage means healthier blood vessels, less arterial plaque, fewer cardiac events. A 2017 study published in the Journal of the American Heart Association found that females are markedly protected from iron overload cardiomyopathy compared to males. In mouse models of iron overload, male and female hearts responded differently. Females showed significantly less structural damage, less functional impairment, and better preservation of cardiac performance under the same iron load. The female heart was not just receiving less iron because of menstruation, it was intrinsically more resistant to iron-induced damage, suggesting that hormonal factors, likely estrogen, provide additional protection even beyond the iron-clearing effect of menstruation. The iron paradox illuminates a pattern that runs through the entire biology of female longevity. The things that look like weaknesses are often hidden strengths. The things that impose visible costs deliver invisible benefits. The things that have been stigmatized, minimized, and used to mark women as inferior are often the mechanisms that keep them alive. Menstruation is inconvenient. It is painful for many women. It limits activity. It requires management and expense. It carries the risk of anemia. These are real costs, real inconveniences, real burdens. But the same process that imposes those costs also maintains the body's iron stores in the range that minimizes oxidative damage, reduces cardiovascular risk, and slows the accumulation of one of the most harmful elements the body stores. The higher body fat that draws cultural criticism is the survival reserve that outlasts famine. The stronger immune system that causes autoimmune disease is the fortress that fights every infection more effectively. The longer post-reproductive lifespan that was dismissed as evolutionary accident is the grandmother effect that may have driven the evolution of human longevity itself, and the menstrual blood loss that has been called a curse in every language ever spoken is the iron-clearing maintenance protocol that the male body never gets. Visible cost, invisible benefit. This is the pattern. And it is the pattern of a body that was designed not for appearance or convenience but for duration, for the long haul, for the 300,000-year answer. Chapter 10. The 300,000-year answer. We have walked now through 10 layers of the female body's survival architecture. We have moved through chromosomes and hormones, through immune cells and telomeres, through mitochondria and fat reserves, through grandmothers and stress responses and the quiet protective logic of menstruation. We have seen the answer from 10 different angles, through 10 different biological systems, across evidence gathered from over 1,000 species, 7 historical catastrophe populations, multiple centuries of demographic records, the oldest humans ever to live, and the most fundamental molecular biology that science can access. And now it is time to step back, all the way back, and see the whole architecture at once. The female body was not built to outlive the male body because of one mechanism. It was built to outlive the male body because of all of them, layered on top of each other, reinforcing each other, compensating for each other's limitations, working together as an integrated system of redundancy, repair, and resilience that the male body simply does not possess. Consider what it means to be a woman at the deepest biological level. Consider what you are carrying right now as you listen to this. You carry 2 X chromosomes. This gives you a genetic backup for over 1,000 genes involved in immune function, brain development, blood clotting, muscle repair, and cellular maintenance. If one copy of a critical gene is defective, the other compensates. Men have no such backup on the X. You carry cellular mosaicism. Different cells in your body are running different genetic programs from your 2 X chromosomes, giving you internal diversity, flexibility, and resilience that no male body can replicate. You are, at the cellular level, 2 different genetic individuals woven into one. A mosaic of strategies distributed across every organ. You carry longer telomeres. From the moment of conception, the protective caps on the ends of your chromosomes were set at a higher starting point than any man's, giving your cells a larger reserve before they reach senescence, giving your organs a longer functional lifespan, giving your body more time before the cellular clocks run down. You carry the estrogen shield. For roughly 35 to 40 years of your life, a molecule was flowing through your blood that repaired your blood vessels, maintained your telomeres, reduced your inflammation, protected your heart, defended your brain, and performed continuous cellular maintenance that the male body, running on testosterone's expensive fuel, never received. You carry the immune fortress. Your immune system fights infection faster, builds antibodies more robustly, maintains immunological memory longer, and survives pandemic after pandemic after pandemic at higher rates than the male immune system. The cost, the risk of autoimmune disease, is real. But the survival advantage is also real, and it has been real for 300,000 years. You carry mitochondria optimized for your body. Every organelle producing energy in your cells was refined by millions of years of selection that tested performance exclusively in females. The males around you carry the same organelles, but theirs were never quality tested in a male body. The hardware is running software optimized for someone else. You carry fat stored in the safest configuration, subcutaneous, readily mobilized, metabolically efficient, a survival reserve that outlasts famine, outlasts cold, outlasts every extreme condition humanity has thrown at the female body across 300 millennia. You carry the grandmother legacy. Your post-reproductive lifespan is not an accident. It is an evolved trait, selected because women who lived longer after menopause had grandchildren who survived at higher rates. Your continued existence past 50, past 60, past 70 is not surplus time. It is the fulfillment of an evolutionary design. You carry the tend and befriend stress response. Your neurochemistry, shaped by estrogen and oxytocin, orients you towards social connection under stress, a strategy that reduces cortisol, lowers blood pressure, dampens inflammation, and produces physiological benefits that the male fight-or-flight response cannot match. You carry the iron paradox. The menstrual cycle that was called a curse is a metabolic maintenance system that keeps your iron stores in the range that minimizes oxidative damage and cardiovascular risk, a cleaning protocol that the male body never receives. No single one of these mechanisms, taken alone, accounts for the 5 to 8 year difference in average lifespan between men and women. But taken together, layered, interlocking, compensating, and reinforcing, they create a system of overlapping protections that has no equivalent in the male body. The male body has its own advantages: greater muscle mass, larger frame, faster sprint speed, higher peak physical output. These are advantages for competition, for predator defense, for short-term survival in physically demanding environments. But they are not advantages for duration. They are not advantages for endurance. They are advantages that serve the first 30 or 40 years of life and then become liabilities, metabolically expensive, hormonally costly, and associated with accelerated aging. The female body's advantages are quieter, deeper, less visible. They do not produce the outward markers of strength that human cultures have traditionally valued: the broad shoulders, the heavy muscles, the deep voice, the physical intimidation. But they accumulate silently across decades into a survival differential that has remained consistent across 300,000 years of human existence. Across every culture, every continent, every historical condition, every level of technological development. 85% of centenarians are women. Over 95% of supercentenarians, those who reach 110, are women. Jeanne Calment of France lived to 122 years and 164 days, the longest verified human lifespan in history. She was a woman. The second longest lived person in verified history was a woman. The third was a woman. Of the 10 longest verified human lifespans ever recorded, every single one belongs to a woman. Yeah, at the extreme edge of human longevity, where every system in the body has been pushed to its absolute limit, where only the most resilient biology can sustain life, the female advantage does not narrow. It widens. It becomes nearly absolute. At 110 and beyond, there are almost no men. There are women. This is the 300,000-year answer. Not a single gene, not a single hormone, not a single clever trick of cellular biology. A system, an architecture, a body engineered layer by layer, redundancy upon redundancy, over evolutionary time so deep it predates our species, for the specific and unmistakable purpose of outlasting the conditions that kill. And the reason evolution invested so heavily in female survival is the same reason that runs through every chapter of this story, from the chromosomes to the grandmothers. The survival of the female was the survival of the next generation, A male who died at 30 had usually already mated. His genes were already in the pipeline. A female who died at 30 may have left behind children who could not yet survive alone. Children who would die, taking her genetic legacy with them. A grandmother who died at 55 may have left behind grandchildren who would have survived with her help, but who will now perish without it. The death of a mother cost more. The death of a grandmother cost more. Not because women are more valuable as human beings. That is a moral question. It is not a biological one, but because in the brutal arithmetic of natural selection, female survival was more consequential for the survival of descendants. And evolution responds to consequences. It builds bodies that survive where survival matters most. And for 300,000 years, the place where survival mattered most was the female body. This is not ideology. This is not feminism dressed as science. This is what the chromosomes say, what the hormones say, what the immune cells say, what the telomeres say, what the mitochondria say, what the fat cells say, what the stress hormones say, what the iron levels say, what the grandmother effect says, what 1,000 species across the entire animal kingdom say, what every country on Earth, in every era of recorded history confirms with demographic data that admits no exception. The female body was built to endure, and it has, and it does, and it will. We began tonight with a question: why do women live longer than men? And we began with the understanding that the popular answers, the behavioural answers, the lifestyle answers, the men take more risks answers, were never wrong exactly, but were never deep enough. They described the surface of the water. They never reached the ocean floor. Now you know what lies at the bottom. Not one mechanism, but 10. Not one system, but an architecture. Not one moment of biological luck, but 300,000 years of relentless, purposeful layer upon layer engineering by the only force that builds bodies, natural selection. Your 2X chromosomes give you genetic backup and cellular mosaicism, redundancy within redundancy, a survival system with its own survival system. Your estrogen maintained your cardiovascular system, activated your telomerase, reduced your inflammation, and performed decades of cellular repair work that no man's body receives. Your immune system fights harder, responds faster, remembers longer, and survives pandemics at higher rates, at the cost of autoimmune vulnerability that evolution deemed acceptable, because the survival benefit was so large. Your telomeres started longer and were maintained better, giving your cells a deeper reserve of replicative capacity. Your mitochondria were optimized for your body by millions of years of maternal selection that never tested their performance in a male. Your fat distribution was configured for metabolic safety and famine survival. Your grandmother legacy ensured that your post-reproductive decades were not wasted time but essential contribution. Your stress response oriented you toward connection, the single most life-extending behavioral pattern in all of public health research. Your menstrual cycle cleared the oxidative burden that accumulates unchecked in the male body. This is what 300,000 years of engineering looks like. Not one brilliant innovation. 1,000 quiet refinements, accumulated over 100,000 generations, each one tilting the odds of female survival a fraction further. And perhaps the most remarkable thing about this answer is how long it was hidden in plain sight. For centuries, for millennia, the female body was treated as the weaker body, the frailer body, the softer body. The body that needed protection because it could not protect itself. The body that was unsuited for the public world because it was subject to inconvenient biological processes, menstruation, pregnancy, menopause, that marked it as less reliable, less stable, less capable than the male standard. And all along, that supposedly weaker body was outliving the stronger one. Every year, in every country, under every condition. The data was there, in the cemetery records, in the parish registers, in the demographic tables, in the centenarian rolls. But the narrative was controlled by the sex that was dying sooner, and that sex looked at the female body and saw weakness, because it measured strength the wrong way. Strength is not only the capacity to lift, it is the capacity to last. And by that measure, the measure that actually determines who is still alive at the end, the female body is the strongest body that evolution has ever built. You were told your body was fragile. It is the most durable biological machine in the natural world. You were told your fat was a flaw. It is a survival system that outlasted every famine in human history. You were told menstruation was a curse. It is a metabolic maintenance protocol that the male body never receives. You were told menopause was the end of your usefulness. It is the beginning of the role that may have driven the evolution of human longevity itself. You were told that aging made you less. But every year you age is a year your body's architecture was specifically designed to reach. Every decade is a decade that evolution invested in. Every birthday past 50 is a birthday that 300,000 years of natural selection chose deliberately to make possible. And you, sitting here tonight, in whatever year of your life this finds you, in whatever state of health or fatigue or strength or grief or quiet contentment, you are carrying in your cells the inheritance of every woman who ever survived when survival was not supposed to be possible, every famine survivor, every plague survivor, every woman who buried a husband and kept the household standing, every grandmother who dug tubers in the hard soil and carried children on her hip, and taught her granddaughter which plants would heal and which would harm. Every mother who nursed through the night and walked through the morning and kept going when every reasonable measure of endurance said she should have stopped. 300,000 years of women who kept going. 300,000 years of a body that was built, layer by layer, chromosome by chromosome, hormone by hormone, cell by cell, to outlast the world that was trying to end it. That is the answer. It has been inside your body your entire life, written in your chromosomes, humming in your mitochondria, flowing in your estrogen, fighting in your immune cells, counting in your telomeres, stored in your fat, running in your oxytocin, clearing in your blood, and no one, not history, not culture, not medicine, not the centuries of men who studied the male body and called it the default, no one can take it from you. Because it is not a belief, it is not an argument, it is not a philosophy, it is what you are made of. It is the 300,000-year answer, and it has been yours all along.